Hello students, I am teacher Svetlana and today we will learn further the components of a eukaryotic cell, that is the lysosome, vacuoles, glyoxysomes, mitochondria, plastids, which are covered in the chapter Cell Structure and Organization. Lysosomes. Lysosomes can be considered as dismantling and restructuring units of a cell. They are membrane-bound vesicles containing hydrolytic enzymes. The enzymes in lysosomes are used by most eukaryotic cells to digest, that is, hydrolyze macromolecules. The lysosomal enzymes show optimal activity at acidic pH. Lysosomes arise from Golgi-associated endoplasmic reticulum. The list of lysosomal enzymes include all types of hydrolases, example amylases, proteases, and lipases. These enzymes are in inactive state and are activated only when a lysozyme comes in contact with another particular organ or the organelle to form a hybrid structure. After the action of the enzyme is over, the lysosome is reformed and reused. Lysosomes are thus found in various structural forms and carry out various functions in the cell. This is a tiny organelle, that is your lysosome. Types. Lysosomes are polymorphic in nature. We can classify lysosomes as primary lysosomes, which are nothing but membrane-bound vesicles in which enzymes are in the inactive state. Have a look at this image here. This is a primary lysosome here. Secondary lysosomes or the hybrid lysosomes, which are formed by the fusion of the lysosome with endocytic vesicles containing materials to be digested, represented as a heterophagic vesicle. This is larger in size than the primary lysosome. Have a look at this image here. This is your secondary lysosome. And then you have the residual body, which is the vesicle containing undigested remains left over in the heterophagic vesicle after releasing the products of digestion in the cytosol. Here, have a look at this image. This is your residual body. Functions. Lysosomes, which bring about digestion of cells' own organic material like a damaged cell organelle, are called as autophagic vesicles or suicide bag. An autophagic vesicle essentially consists of lysosome fused with membrane-bound old cell organelles or organic molecules to be recycled. Remember, every week a human liver cell recycles half of its macromolecules. Have a look at this image here. This is your endoplasmic reticulum, this is the Golgi apparatus, and this is the lysosome, which is in close proximity to it, and this is where it is engulfing a damaged organelle and digesting it. It does this with food also. When food enters into the cell, it eventually gets digested because of lysosomal activity. So this lysosomes bring about intracellular and extracellular digestion. The intracellular digestion is brought about by the autophagic vesicle or the secondary lysosomes, which contain foreign materials brought in by the process like phagocytosis. Food vacuoles in the amoeba or macrophages in human blood that engulf and destroy harmful microbes that enter the body. Then you have extracellular digestion, which is brought about by release of the lysosomal enzymes outside the cell. Example, we have the acrosome, a cap-like structure in the human sperm, is modified lysosome which contains various enzymes like hydroolidase. These enzymes which bring about fertilization by dissolving the protective layer of the ovum. Have a look at this image here. This is the acrosome, which has the hydrolytic enzyme. When it comes in contact with the protective layer of the ovum, it releases the enzyme and digests this protective layer to enhance the process of fertilization. During metamorphosis processes found in many organisms, lysosomal enzymes help in reusing the tissue of the reductant organs. So they also help in destruction of malpighian cells. Malpighian cells are cancerous cells. Example, we have the T lymphocytes. Vacuoles, they are large membranous sacs found in the cytoplasm. Have a look at this image here. This is your vacuole. These store substances that are not necessarily useful for the cell, like water, sap, excretory product, and other materials. Plant vacuole consists not only water, sugars, and salts, but also contains pigments and toxic molecules, and also occupy up to 90% of the volume of the cell. The vacuole is bounded by a single membrane structure known as the tonoplast. Here, this is the tonoplast. which in plant cell facilitates the transport of materials and some ions against the concentration gradient inside the vacuole. Thus, the concentration of the material is tend to be at higher levels in the vacuole than to be in the cytoplasm. Animal cells also have vacuoles, but they have much prominent in the case of plant cells. So if you see, the animal vacuole is quite tiny as compared to the plant vacuole, which is large in shape. Thus, plant cells have typically large central vacuoles filled with watery fluid that gives added support to the cell. Vacuoles store excretory products or even compounds that are harmful or unpalatable to herbivores, thereby the plants. Attractive colors of the petals are due to the storage of such pigments in the vacuoles. So let's have a look at the types of vacuole. Intake of food or foreign particle by phagocytosis involves formation of food vacuole. In freshwater unicellular forms like paramecium, excretion and osmoregulation takes place by contractile vacuoles. These vacuoles maintain the turgidity of the cell. In addition to the endomembrane system, there are several other cell organelles bound by single layer of plasma membrane in the cell. These are called as microbodies. These are found in both plant and animal cells. They are minute membrane-bound sacs. Microbodies contain various types of enzymes, and based on which they are classified into different types, few of which are explained here. We have spirosomes. They are found mainly in the cells involved in the synthesis and storage of fats. Example, endosperm of oil seeds. The membrane of spirosome is half unit membrane, that is, this membrane has only one phospholipid layer. Peroxisomes. Peroxisome contains enzymes that remove hydrogen atom from substrate and produces toxic hydrogen peroxide by utilization of oxygen. At the same time, peroxisome also contains enzymes that convert toxic H2O2 to water. Conversion of toxic substance like alcohol takes place in the liver cells by peroxisome. This is how a peroxisome looks. And this is the general structure of the microbodies. They have a lipid bilayer membrane, and inside they will have the different enzymes within it. Glycosomes are also a type of microbodies. The membrane-bound organelles contain enzymes that convert fatty acids to sugar. They can be observed in cells of germinating seeds, where the cells utilize stored fats as source of sugar till it starts photosynthesizing on its own. So here is how the glycosome looks. The next important organelle is mitochondria. They are membrane-bound cell organelles essential for aerobic respiration of the eukaryotic cells. They are also known as the powerhouse of the cell. Thus, they produce cellular energy in the form of ATP. Occurrence. Mitochondria are present in all living cells except the prokaryotic cells and certain specialized eukaryotic cells such as anaerobic cells and mature RBCs. It is revealed from the studies that mitochondria is not easily visible unless it is specifically strained. Let us have a look at the ultrastructure. That is how the mitochondria appears under the microscope. A mitochondrion contains two membranes, that is an outer membrane and an inner membrane. The outer of which the outer membrane is smooth and forms the continuous boundary of the organelle. The inner membrane is semi-permeable to some metabolites. It is enfolded into the matrix as incomplete partitions called as cristae. Here you can see the inner membrane is folding, and each of this folding is called as a cristae. The cristae are responsible for increasing the physiological active area or the surface area. The density of cristae determine the intensity of respiration. The outer and the inner membrane divides its lumen into two aqueous compartments separately, that is the outer and the inner compartment. The inner compartment is also called as matrix. Here, this is the inner compartment and it is called as matrix, which forms the inner core of the mitochondria. The matrix also possesses single circular DNA molecule, a few RNA molecule, ribosome of the 70S type, and which are the components required for the synthesis of proteins, lipids, and various enzymes of the Krebs cycle and other pathway. The mitochondria divides by fission. The two membranes of the mitochondria have their own specific enzymes associated with mitochondrial functions. Oxysomes. The inner membrane of the mitochondria bear numerous particles called as oxysomes. F1, F0, they are also called as Fernandez and Moran elementary particles, mitochondrial particles. So there are different names to it. Each particle consists of a head region, a stalk region, and a base or a foot region. Head is called as the F1 or the lollipop head, faces towards the matrix, and the foot is embedded in the inner membrane. Can you see this image here? You can see that the head is present outwards and the foot is embedded in the inner membrane. Head acts as an enzyme ATPase synthesis and foot as a proton channel. Oxysomes are involved in proton pumping and ATP synthesis. So this is the site where ATP synthesis takes place in the mitochondria. Plastids. These are semi-autonomous organelles that have double membrane envelope. Plastids have their own genetic material, that's the DNA. Due to their large size, they can be easily seen under the microscope. Now, plastids are found in all plant cells and euglenoids except in some protistins. Type. Plasmids are differentiated into 3 different types on the basis of their color, that is the type of pigments found in them. The first is the leucoplast. These are colorless plastids of various shape and sizes which store nutrients in the form of carbohydrates, lipids, and proteins. For example, amyloplasts are the carbohydrates or starch containing leucoplast. Example, rice, wheat, potato. Amyloplasts are larger than the normal or the original size of the leucoplast. We have elaioplast, which store oils and fats. Then we have areoplast, which are protein storing leucoplast. Example, maize. Chromoplast. These are the leukocytes which are yellow or reddish in appearance because of the presence of fat-soluble carotenoid pigments called as carotenes. Xanthophylls and some other pigments are also present as the fat-soluble carotenoid pigments other than the carotene. Example, orange color of the carrot. Here are some types of plastids for you to refer. Then the last and the important type of plastids are the chloroplast. These are the plastids which are greenish in color containing photosynthetic pigments, chlorophyll and carotenoids. These pigments are responsible for trapping the light energy essential for photosynthesis, that is the synthesis of organic food from an inorganic raw material in the presence of sunlight. Chloroplasts occur in major numbers in photosynthetic mesophyll cells of leaves and green stem. Their shape is lens-shaped, oval, spherical, discoid, or ribbon-like organelles. They also have variable lengths from 5 to 10 mm and width from 2 to 4 mm. Their numbers vary as per the cells. For example, we have Chlamydomonas, a green algae, which will have 2 to 40 per cell in the mesophyll. In its infrastructure, chloroplasts are bounded by double membrane envelope like mitochondria. The two membranes are smooth and are thick of about 90 to 100 angstroms. The inner membrane of the chloroplast is less permeable than the outer one. The inner membrane is grounded by a space known as stroma or matrix. This is the stroma, which has a dense, colorless or granular substance mainly formed of soluble proteins. It also contains enzymes which are essential for the synthesis of carbohydrates, lipids, and proteins. Thylakoids are a number of membranous flat structures that run throughout the matrix of the stroma. With several thylakoids are arranged or organized in stack, they appear like piles of coin, which are called as grana. This here is a grana. Many flat membranous tubules interconnect the thylakoid of different grana known as stromal lamina. So here they are all interconnected. What are the major functions of your chloroplast? They help in photosynthesis, that is formation of organic compounds. They help in consumptions of CO2 and release O2 during photosynthesis. May also change into chromoplast in order to provide color to different flowers and fruits. They help in storing fats and lipids. They function in transduction of energy. Thank you students for participating in this lecture. See you soon again with the next topic. Till then, take care, be safe and keep learning.